First of all, thanks very much, Steve, for the kind introduction. Thank you for the invitation to speak today. It's really exciting to see an interdisciplinary workshop in this space. And hopefully some of what I tell you today will actually illustrate how we're approaching this from a multidisciplinary angle. These are my disclosures. So in the next few minutes, I really want to just show how we've gone about understanding cancer evolution to date. And then in the latter part of the talk, I will focus on a more recent insights on spatial dynamics of clonal evolution as relevant to metastases. And in fact, we work largely, our work is based on patient data, and arguably these data are really critical as a starting point to provide the insights into clonal dynamics at the scale that's really relevant to the disease. These data also help us to then derive parameters that are helpful for mathematical modeling, which can give us more repeatable predictions of cancer evolution, and therefore they can help us understand future growth and facilitate prognostication. And of course, a lot of this is about genotypes, but through model systems, we're able to separate out the phenotypic effects of these different genetic lesions individually or in combination, and then test the range of contexts in which this fitness can be altered in the context of tumor microenvironment or therapy. And then finally, uncovering and quantifying these laws of evolutionary dynamics will not really be sufficient to bring this understanding into the clinic. So one of the sub-programs in the lab is really focused on how we put these insights into clinical use through more tractable methods. But today, really, I'm just going to focus on these two settings. We work on two cancers in the lab, melanoma and kidney cancer, but I will only focus on the latter today. And arguably, renal cancer really has the necessary diversity of metastatic phenotypes to help us understand how this might be underpinned by different evolutionary trajectories. So some tumors which are not shown in this schematic here actually are detected at very early stages. They never progress, they never metastasize. There are tumors that following curative resection of the primary tumor metastasize very late, sometimes decades later, and they have a predilection for endocrine organs, which is curious in itself, and they have an excellent outcome following the resection of these solitary metastases. Now, the tumors that spread earlier in the course of disease following the resection of the primary tumor do so either in a sort of limited fashion, exhibiting an intermediate metastatic efficiency. So often they will relapse 2 or 3 years after the primary tumor is removed, but will be confined to a single organ, either as a solitary metastasis or what we refer to as oligometastases, so a small number of low volume metastases. And unfortunately, there is this group of patients where very quickly after surgery to the primary tumor site, there is very efficient, rapid spread comprehensively throughout the body of metastases, and these patients have a very short prognosis. And this certainly suggests that there is presence of occult metastases at the time when the primary tumor is discovered, but these are not detected radiologically due to sensitivity issues. And then the other reason why this is a compelling model to understand cancer evolution is because the kidney cancer genetics are very stereotypical. There's only a handful of mutations in these 4 tumor suppressor genes, which really help us to explain a lot of what happens in kidney cancer. So I won't spend too much on this because this is published data, but it really sets the background to the ongoing work in the lab. So through spatially aware clonal profiling, so this is multi-regional profiling of primary tumors, we've established over the last few years in the context of the TracerX renal program that there are 3 distinct modes of clonal evolution that are relevant to clear cell kidney cancer. So this is the most common subtype of kidney cancer. So slow-growing tumors are characterized by dominance of a limited fitness clone, and we inferred this limited fitness through the observation of very long time of growth between the emergence of the most recent common ancestor and diagnosis, as well as the fact that these tumors grow to a very small size usually. There is no detectable ongoing evolution of these tumors, although, of course, this might be a result of limits of sensitivity as well with bulk sequencing. There are these slow-growing larger tumors which have very high levels of clonal complexity. And then lastly, there are fast-growing large tumors where the founding cell or near founding cell is at its fitness peak. This leads to a clonal sweep, and there is lack of fixation of any other populations. And really, the fitness in this case appears to be driven by acquisition of high levels of chromosomal complexity and fixation of multiple aneuploidies, either subclonally in these more Darwinian branched evolution patterns, or clonally, where in the context of this clonal sweep. So coming back to those clinical phenotypes now that I mentioned to you earlier, we know that the primary tumors in this group grow to a large size, actually comparable size to these tumors, but just over much longer periods of time. So median here of 25 years and a median of 4 years here. And intriguingly, although these patients present with metastatic disease, it's often with a gradual course, starting at a single metastatic site. And what's very relevant from a clinical perspective is that these were the patients that we identified benefit from the removal of the primary tumor, even following the diagnosis of metastatic disease, suggesting that ongoing evolution at the primary site might further feed metastatic potential of that primary tumor and lead to ongoing metastatic dissemination. And similarly, resection of these single metastatic sites sometimes can lead to cure in these patients, if not a prolonged survival without active metastatic disease. However, in this context of punctuated evolution, again, where there is evidence of a clonal sweep, there is rapid metastatic progression, and these patients don't benefit from surgery to either the primary tumor or the metastatic sites, and indeed they have a very short prognosis. But in both cases, metastasis-competent clones are very similar genetically. And here we were able to do something that was alluded to in the last talk, which is to really compare matched primary and metastatic tumors. And furthermore, because we have such refined clonal resolution, we can directly compare and contrast the clones that do not metastasize, represented here in blue or red, with clones that do metastasize, represented here in brown. And what we see is that they are characterized by high levels of copy number alterations and aneuploidy. There is no difference in the tumor mutational burden or burden of driver mutations. And they're specifically enriched for chromosomal arm losses at 9p and 14q. So this is quite intriguing because it suggests that the timing of the acquisition of metastatic competence is highly relevant to how metastases present and manifest. And this may simply reflect sort of a numbers game, if you like, where when most cells at the primary tumor site are metastasis competent, this leads to comprehensive metastatic spread, efficient metastatic spread. However, when this metastatic competence is really limited to a subclone at the primary tumor site, then the process is inefficient. However, we can't exclude the possibility that, for example, the clonal interference in these clonally complex tumors also bear relevance, and also that these clones may be phenotypically different in these different contexts, even though they're genetically identical. So the exception to this rule were pancreatic metastases, and you'll remember at the start I told you that pancreatic metastases tend to present very late following the removal of the primary tumor, and that they behave fairly indolently. They're often assumed to be dormant. So interestingly, these clones actually derive from the ancestral clone. So this is the least fit clone at the primary tumor site. And this may well explain both the indolent behavior of these metastases, but also the lack of cross-metastatic seeding. So when these pancreatic metastases are detected, they're often seen in isolation as opposed to in the context of wider metastatic spread. So these data motivate then further application of this type of framework, and I hope I've convinced you that it is a useful framework relevant to the human disease. And we're currently conducting these analyses across a larger cohort of patients, where we're also exploring the contribution of epigenetic changes to this evolutionary classification. But this is all ignoring what happens to clonal diversification and for metastasis formation in the spatial context. And we took advantage of one of the unique aspects of our clinical cohorts, which is that these spatial sampling maps can be combined with molecular data to map the exact locations of individual clones. And that can reveal how genetically distinct subclones grow and evolve spatially at the primary tumor site. And this allows us to do a whole range of analyses, such as a correlation between genetic distance and spatial geographical distance. This can be a surrogate for clone fitness, for example. We were particularly interested to understand the locality of metastasizing clones and also to interrogate whether clonal expansions were contiguous in nature or whether there was evidence of clonal migration. So in this exercise, we examined the differences between the tumor center, here in lighter pink, and the tumor margin in brown. And we observed that the clones which are located in the tumor center, on the whole, have higher levels of genome instability, higher levels of driver somatic copy number alterations. They have high levels of proliferation, as reflected in Ki67 here. They're more likely to exhibit necrosis, and they're more likely to have high Fermat grade, so 3 or 4. And furthermore, we find that metastasizing clones, as I've shown you, we're able to pinpoint which are the metastasizing clones because we have the matched metastases from these patients, so we know which are the founder clones. And where in the primary site they've emerged, and we see that they're more likely to originate from the tumor center, although not exclusively so. Assessing the relationship between the spatial and the genetic distance, we find that the largest subclones were dominated by copy number alterations as opposed to driver mutations, suggesting that they have a very large selective advantage, even though copy number events tend to occur later in the evolutionary timeframe relative to mutations. And finally, when we modeled the selective pressure imposed by necrosis in these simulated tumors, we observed that presence of necrosis facilitates expansion of copy number alteration driven clones. So putting all this together, it seems that there is a model of accelerated evolution in the tumor interior, where perhaps harsh hypoxic environmental conditions lead to necrosis, and then there is a greater opportunity for driving genome instability and therefore fixing these clones that have high levels of copy number aberrations, which then afford metastatic competence. But these cause and effect relationships are really inferred at this stage, and we're currently investigating them functionally. And furthermore, to say that the tumor necrosis, which is observed in clear cell kidney cancers and carries prognostic information, can be either acute or chronic, and we're certain that that also plays a role in these cause and effect relationships. And then finally, I mentioned that we were interested in how these clones spread at the primary tumor sites. So we see that majority of the clones that we detect actually expanded contiguously, and very, very few showed evidence of dispersal. And I don't know whether you can appreciate that on this slide here, but for example, this green clone down here doesn't have a contiguous pattern of growth, but instead jumps across another clonal outgrowth. And only in this instance, and as I said, it's relatively rare, certainly in our cohort, it actually coincides with the metastasizing clones, suggesting that potentially this pro-migratory phenotype selected for at the primary tumor site also affords metastatic potential. So then, just in the last few minutes, I wanted to go back upstream to evolution of the primary tumors and what we've done in terms of understanding how the spatial patterns of tumor growth might impact subclonal diversification. And as you can see, as I've shown you before, these are highly relevant to emergence of metastatic potential. So there was a keen interest in understanding how branched evolution occurs as opposed to how punctuated evolution occurs. And for this purpose, Zhao Fu had developed a model to simulate evolutionary dynamics in clear cell kidney cancer. The fitness parameters were taken from the primary data that I've shown you earlier. And Zhao was particularly interested in understanding and contrasting surface and volume growth. I would invite you to read Zhao's paper on Research Square, but I just will highlight a couple of main findings as relevant to this talk. What Zhao observes is that the tumors that evolve, that grow specifically at the surface tend to grow by branched evolution. So there is subclonal diversification very early on, but he also notes there is this change in tumor morphology. So these tumors have many budding structures at the surface, and contrast that with these tumors that grow by volume growth. So here, in this model, it's assumed that every cell in the tumor is actively proliferating as opposed to just those on the surface. And these tend to coincide with a punctuated evolutionary pattern. So there is a rapid clonal sweep when an appropriately fit clone emerges. And as you can see, the outline of these tumors is relatively smooth. So does this bear any relevance to what we see in patients? Well, we were able to, again, thanks to the highly annotated clinical cohorts we're able to go back to the patient data and specifically radiological images. And before doing that, Zhao essentially rewound these simulations and observed that these budding structures that were shown in the previous slide actually precede subclonal outgrowth. And indeed, when we looked at very early stage clear cell kidney cancers, so these are the ones that hadn't manifested progression or metastases, we could see that in some of these, there was evidence of these early budding structures. And when we then correlated them to the spatial maps of how the tumors were sampled, we could overlap them with areas of very recent subclonal diversification. So this really raises the possibility that evolutionary potential could be predicted from routinely acquired clinical data, which goes back to my original point of how we translate this evolutionary understanding into the clinic. And therefore, monitoring of early stage tumors radiologically could help us understand whether they're destined to diversify or indeed metastasize. So I'll finish up with conclusions. So I've shown you that in the context of clear cell kidney cancer as a model of cancer evolution, we observe that gradual evolution or more Darwinian evolution results in slow growth and attenuated metastases, and that this bears relevance to the clinical management of these patients. That punctuated evolution with early fixation of chromosomally unstable clones leads to rapid growth and metastases that perhaps simply reflects the efficiency of metastatic spread from a primary tumor that's dominated by a metastatically competent clone. And I've shown you just in these last couple of slides that mode of evolution may well reflect the patterns of tumor growth. So where the growth is predominantly at the surface, this coincides with branched evolutionary patterns where the growth happens throughout the tumor, the so-called volume growth. This is more likely to reflect punctuated evolution in a clonal sweep. And I really want to highlight this point. We very much see that metastatic competence emerges at the site of the primary tumor. And this is in contrast to some other examples of parallel evolution of metastatic potential, where early clones derived from the primary tumor may continue to evolve outside the primary tumor, sometimes in places of immune privilege and so on. And therefore, there is a great deal of divergence between the primary tumor and its metastases. But certainly in the context of clear cell kidney cancer, we're confident that metastatic competence emerges at the site of the primary tumor. Whether it emerges early or late is very relevant to the manifestation of metastatic disease. And I've shown you that the conditions in the center of the tumor seem to favor selection of metastatic competence. And this metastatic competence, certainly, again, in this setting, seems to be underpinned by chromosomal level events rather than gene mutations. And this perhaps makes sense considering, again, shown beautifully in the previous talk, the entire metastatic cascade and the many different phenotypes that are required for metastases to form. That chromosomal level events, which will affect the gene expression of hundreds of sites, may well be what's required to achieve that multitude of phenotypes. And with that, I will end and thank everyone who's been involved in this work, but really most of all our patients and their families who really facilitate these insights. And if you're excited by this research, please do get in touch for opportunities in our lab. Thank you.
Samra, thank you so much for a really outstanding talk. One of the elements of the talk that particularly excites me as a physicist and mathematical modeler is the increasing emphasis on constructing mathematical or computational models of the tumor dynamics. And you've given a wonderful example of the emergence of budding structures and how that can be predictive of the subsequent trajectory, metastatic trajectory. Are you doing similar models of the sort of signaling pathways that might be implicated in some of these dynamics?
Yeah, so we're deliberately have kept the model extremely simple at this stage, and have used the parameters that we've derived from the primary data to infer fitness. And of course, you know, I'm very mindful of the fact that we're only inferring that based on genetic correlations at the moment. There is a lot in the tumor microenvironment, of course, that we can model. There is a lot in the signaling pathways that we can model. Kidney cancer doesn't lend itself particularly well to this because it's not an oncogene-driven cancer where the signaling cascade would be sort of a straightforward readout, if you see what I mean. So all these genes that I showed you at the beginning that are located on 3P, they're tumor suppressor genes, they're chromatin modifiers. They have such a wide range of downstream effects. That untangling that really takes us back to the preclinical models. But that's what's going on in the lab at the moment through genome engineering. And we hope to eventually feed those very pure fitness effects into the models as well to allow for a more complex understanding that can recapitulate what we see in patients.